In this video, we explore and demonstrate the gram stain, a common differential staining technique used to distinguish between gram-positive and gram-negative bacteria. Here, we are labeling 4 slides that will each have a smear prepared on them, one for E. coli, one for S. aureus, one for B. cereus, and the final one a mixture of 2, S. aureus and E. coli. Before we can begin the gram stain, we must first prepare a smear and fix cells to our slides. Notice that I wrote in a Sharpie marker on each of these slides, but during the gram stain procedure, we will use alcohol that will wash it away. Therefore, we will prepare the smear on the opposite side. As was done in the simple staining lab, in order to prepare a smear, we must first sterilize our inoculating instrument, and then grab the cells from the appropriate medium. Now, in your lab manual, it states that you would have or be given an agar slant. In this video, I'm using a nutrient broth medium. The difference is that when using an agar slant, you would first put a drop of water on the slide to help disperse and spread out the cells. But in this case, we're using a broth and no drop of water added to the slide is necessary. E. coli was just added to the slide, and now we're going to wait for that to air dry. While that is drying, we're going to grab our second culture of Staph aureus, which is also in a liquid broth medium. Once again, we will, of course, sterilize our loop and spread the cells on the slide, always in the size of roughly a dime to a quarter. We will repeat this for the next 2 slides to prepare our final 2 smears. Because the broth is an aqueous solution, that is, based in water, it does take a few minutes for the water to evaporate. But once it does, you should see a smear like in this slide. I recognize it's a little bit difficult to see in this video, but hopefully you see that smudge or residue, which represents the cells left behind on the slide. This one recalls the mixture, including Staph aureus and E. coli, and you can see an especially large smear on that because 2 loops of cells were used. Anytime you prepare a smear for the purposes of staining, you must first fix the cells to the slide. In this case, we will use 95% methanol, which is methyl alcohol, to do our fixation. In earlier videos, you may have seen heat fixation, which is done by passing the slide back and forth through a Bunsen burner flame. But just as well, we can flood the smear in methanol and let it soak until the methanol evaporates. Remember, by fixing cells to the slide, you kill them and adhere them so they don't wash away when you do the subsequent staining steps. Once our smears are prepared and our cells are fixed to the slide, it's time to begin the gram stain. A gram stain begins much like simple staining as demonstrated in earlier videos. In simple staining, we employ any one of a number of basic stains to stain typical bacterial cells. But in a gram stain procedure, we always begin with the same basic stain for our primary stain. That is crystal violet. So again, this first step is called the primary stain because it's one of 2 stains that are going to be used here. Now, just like before with our simple stains, you'll flood the smear, and it's okay if the stain drips over the sides of the slide. The slides are suspended on a rack above a trap that will catch any excess stain. Furthermore, remember that you fix the cells to the slide, so the stain isn't going to carry cells with it into that trap. Once your smears are soaked in crystal violet, you must let it sit for 1 minute. After 1 minute, it's time to wash off each of the slides with water. Tap water is acceptable for use here. However, you want to gently rinse your slide with water. Therefore, you wouldn't want to put your slide under a strong faucet. Notice how I drip the water down an angled slide. And also, it's okay if some stain is remaining at the bottom of the slide. Continue rinsing off each of your smears before moving on to the next step. After the primary stain has been rinsed, we move on to the second step of the gram stain procedure, which is to use a mordant. Specifically, the mordant used in a gram stain is Gram's iodine. Once again, we're going to flood each of our smears with the Gram's iodine. The Gram's iodine is going to intensify the stain, but also form a complex with the crystal violet that plays a role in the differential staining of gram-positive from gram-negative cells. Let the iodine sit for 1 minute. After 1 minute of soaking in Gram's iodine, we're once again going to rinse each of our smears with water, letting the iodine run off the slide into the trap below. Again, you don't want to do this forcefully, so avoid using a strong faucet and use something like a dropper bottle to rinse your slides. Our third step of the Gram stain is perhaps the most critical step, and that is the use of ethyl alcohol, also known as ethanol, to decolorize our stains. Now at this point, whether the cells are gram-positive or gram-negative that we're staining, they're all stained with crystal violet. It's during this step that we accomplish the differentiation between gram-positive and gram-negative cells. As I put the ethanol on the slide, notice that I'm dripping it 1 drop at a time down an angled slide, and though it's difficult to see in this video, I'm keeping an eye out for whether or not the alcohol is running clear. If I still see that blue or violet tint to the liquid as it runs down the slide, then I continue adding more drops of alcohol. Once I do not, and again it's running clear, I stop and move to the next slide. The reason this is critical is because the ethanol is washing the crystal violet away from cells, and it has the potential to do that to both gram-positive and gram-negative cells. However, gram-negative cells, having far less and thinner peptidoglycan in the cell wall, differentially washes out the crystal violet much faster than a gram-positive cell wall would. So if you apply the ethanol correctly, in other words, not too much, you will only decolorize the gram-negative cells and leave the gram-positive cells stained violet with crystal violet. Gram-negative cells will appear colorless after this step. Once again, after the decolorizing step, we'll need to do another wash with water. Tap water is acceptable. Make sure to go through all 4 slides, washing off the alcohol that was added to each. Our final phase of the Gram stain is the counter stain, and for this we use the basic stain known as safranin. Safranin stains a light pink color. It will be added to each slide. Remember, you don't know which ones are gram-positive and which ones are gram-negative. However, the gram-positive cells that are still stained with crystal violet will not show this light pink color. Meanwhile, the otherwise colorless gram-negative cells will take up the stain and therefore appear light pink when we view them under the microscope. We must allow the safranin to sit for 45 seconds, and after 45 seconds, we're going to do one final wash with water. Again, tap water is acceptable. Thoroughly wash off the safranin from each slide. Finally, before we view the slides on the microscope, we must dry them off. You may use bibulous paper for this, or you may simply use a paper towel, as I'm doing here. Notice I fold the paper towel over the slide to blot it dry. I'm not scrubbing the slide, as I don't want to mechanically remove the cells that we've otherwise fixed to the slide. Finally, we get to view our specimens under the microscope. First is E. coli, which is pink and rod-shaped. Then we have our slide of B. cereus, which is rod-shaped but purple. Then we have our Staphylococcus aureus, which is clustered, spherical, and purple. And finally we have our mixture of E. coli and Staph aureus that looks just as in the prior images.